Welcome to Derms on Drugs, a video podcast brought to you by Scholars in Medicine, the best platform for education in dermatology and provided at no cost to medical providers. Derms on Drugs is where cutting-edge derm meets hit-or-miss comedy. I'm Matt Zires, and each week I'm joined by my residency buddies, Doctors Laura Ferris and Tim Patton, where we use our 60 years of combined derm experience to discuss, debate, and dissect the hottest topics in dermatology. It is everything you need to know to be on the cutting edge, and you'll actually have some fun listening. New episodes drop every Friday on Scholars in Medicine, Apple Podcasts, Spotify, and any other major podcast platform you might use. And just a reminder that this is a video podcast, so if you want to see the key figures from the tables, they're in the video part, and then on the Scholars in Medicine platform, the links to all of the articles we talk about are there. So this week, we've got one of our patented six-pack episodes where we are going to go through what has been the coolest, most interesting stuff we've seen in the literature over the last few weeks. And we are going to start off with Dr. Ferris. Dr. Ferris, what do you got?
All right. Thanks, Cyrus. So I have a paper from Lancet Rheumatology, Hagen et al., local immune effector cell associated toxicity syndrome in CAR T-cell treated patients with autoimmune disease, an observational study. So that's a lot. So I picked this because it's kind of interesting. It's a newly described chemotherapy or, drug toxicity. So we are going to call this LICAT. Okay. So this is an observational study of 39 patients.
Ferris, wait, I got to stop you first just because I'm too far behind to even start here. What is CAR T? What is the therapy here? Or are you going to get into that?
Well, so the therapy are these modified T cells that are engineered to target CD19. Okay, so instead of having like an antibody that depletes CD19 T cells, instead of having an antibody that depletes CD19 positive B cells, you have this modified T cell that does the same thing. Now, this is sort of CAR T cells were originally developed to treat like hematologic, mostly malignancy. So it's basically an engineered T cell that attacks, in most cases, a tumor cell, but they've been engineered to actually attack CD19 positive cells, which are going to be B cells. So these are T cells. I mean, this is like for the worst of the worst autoimmune conditions.
Do they like take the patient's T cells out, genetically modify them, and then put them back in? Or are these just some random person's T cells?
No, it's not some random person's T cells. They are your own T cells.
That would be slightly dangerous.
It would be a little bit dangerous.
Versus host may be a problem.
I should have specified.
Okay, all right. So
taking that for granted.
So this is almost the equivalent of rituximab, but instead of an antibody, we're giving people cell-mediated, which should be denser and more effective. Is that in longer
Yes, it should be more targeted, more effective. And so interestingly, because it's relevant to this paper, what they say these are better at penetrating into tissues and depleting B cells within tissue. And that's why they think they're actually better at treating autoimmune disease. So you harvest the patient's T cells, you engineer them to react to CD19. You then basically cytodeplete these patients, right? So you give them like Cytoxan and fludarabine, and then you give them back these modified T cells. I mean, this is like serious business. This is not something we're going to start doing for every disease, but it is done.
And I assume when they use it for cancer, I assume that they can target to some antigen that is specific for that cancer.
Yes, they could. It's not all CD19, so you can sort of modulate these to be, specific to whatever disease you're treating. Patton, am I saying that all right? Is there anything you'd modify to what I'm saying?
No, that's right. Except, I mean, isn't 90% of CAR T cells like CD19's the target? or am
I think that there are more. So CD19 targeted CAR T cells can be used to treat B cell malignancies too. But there's also been like CAR T cells that have been proposed for treating other autoinflammatory diseases or other malignancies.
I guess I'm thinking of the ones that are commercially available. I assumed it was only the 19s. I could be wrong though.
Okay. Yeah, I have read about them in studies. I got to be honest, I've never had to look at doing this for a patient. So,
And this is different than tumor infiltrate, because in cancer
yeah,
they talk also about tumor infiltrating
this
lymphocytes.
is different than TILs. TILs
Okay.
are your own tumor harvested. You take out the cells, you grow up and activate the cells and give those back. So those
Okay.
are going to be like polyclonal. So this is your own T cell modified to attack CD19.
All right. Okay. I'm all caught up
Okay.
now. Now I'm ready to hear about the
Now
paper.
you're ready.
Okay.
Okay. So this is looking at using these not to treat malignancy, but to treat like bad autoimmune disease. Observational study, 30 patients, 20 had SLE, 13 systemic sclerosis, 6 had idiopathic inflammatory myopathy. Like 2/3 were women, as you might expect. So what they described these local reactions, which they called LICATS, which is local immune effector cell associated toxicity syndrome. Okay, so the reason I picked this is just like this isn't something that I think about when I think about toxicities to immuno or chemotherapy, to therapies or to any drugs. So they found that 77% of patients were affected by LICATs, with a median time of onset of about 10 days. Now, this differs from when you give CAR T cells, you think about something called cytokine release syndrome, which is basically like massive T cell activation in your tissues. So you get, massive inflammation, hypotension. These patients can get pretty sick. This is differentiated from that. So what they found is that this occurs exclusively during the B cell aplasia phase. So basically, when you have all your B cells have been wiped out and you've got no B cells, that you get this reaction. So, and it's only involves the organs that were affected by the autoimmune disease. So,
Oh,
yeah,
that's
so it's
cool.
really interesting. So, like if you had cutaneous manifestations of your disease, i.e., the SLE patients, you got skin toxicity. If you had lupus nephritis, you had renal toxicity. If you had myositis, you had, if you had myopathy or myositis, you got muscle disease. So, and most of these were actually like relatively mild. They went away with some prednisone, and only a couple were significant. And so if you compare this, you'd be like, okay, well, we do this with like Rituxan or CD19 targeting antibodies, and it turns out that you don't get this. And the authors argue this is because they don't really deplete the B cells deeper in the tissue, and that this is something unique to the fact that you deplete the B cells, and that is driving this immunologic resetting and triggering a stable remission, but that in the process of this resetting you can get this autoinflammatory.
All right. Wait. It seems more intuitive to me that when you give the antibody just attaches to the bad cell and kills it. But this, you're giving these cells that they then like dock up and like release stuff that causes an inflammatory reaction that you wouldn't get with the antibodies. Why is
Yeah.
that? That seemed like
Yes, I think so. That is one, potential. But the thing that's interesting is when they see this, the B cells are actually gone. So it's not like, oh, look, here's a T cell, B cell conjugate. Like there's no B cell, present in the tissue at the time that they like it happens after that. So that's interesting. Now, the other thing, like later in the paper, when they talk about what could this be, that they do talk about is that you're going to also be reinfusing some of the patient's own T cells. And it may just be like, hey, now you've given them Cytoxan and fludarabine, and now you're actually, putting back some of the pathogenic T cells.
so you're almost getting a little graft versus host disease.
Yeah, sort of, except
No,
it's
no,
like
no, not
host
really. Yeah,
versus
yeah, yeah.
host.
No.
Yeah.
Okay.
Okay.
Huh.
So yeah, interesting. So most common things that they saw were transient worsening of renal function, proteinuria, transient skin rashes, and then musculoskeletal. So if you have bad lupus arthritis, like you would get a little flare of like a transient musculoskeletal flare too. So I thought that was sort of interesting. So of the skin, 19 so they had 19 patients with skin manifestations. So it was like an erythematous rash, swelling of oral mucosa, mucositis in one. There's one that had these cutaneous nodules on the hands, one who had worsening of digital ulcerations. And the median time for skin manifestations was like 19 days. So median time to see LICAT start at all was about 10. Median time for skin was about 19. Most of them did not need anything more than like prednisone treatment. They did have a couple patients that had skin biopsies. It's interesting because it's a rheumatology patient paper, and they're like, and there was kind of like not much inflammation. It was nothing. But then when you actually read what they said, in 3 patients, skin biopsies were performed. So they had one patient who had a biopsy day 30 after getting their CAR T cells, and they saw interface dermatitis infiltrating CD4 and CD8 T cells, and then myeloid cells as well. They had another one that was taken on day 70. They just saw some like CD123 positive myeloid cells, no B cells. And then another one that was taken on day 35, and they showed like CD4 and CD8 positive T cells and not B cells. So at the time of biopsy, there was like really like B cell depletion in the peripheral blood, no serologic signs of lupus, like no double-stranded DNA that was negative. CD3 and CD4 were also normal. So, just interesting because I think like we will potentially start getting called for this. I think it's something for us to be aware of. And they had some good pictures, one of oral ulceration, the manifestations were not necessarily that dramatic. It was sort of that like, erythematous papules on the face. There was a couple pictures that were of patients who had like what looked like these petechial lesions on the fingers. But, after prednisone treatment, they all seem to go away. They also have like a CT scan of the lung. So I thought, this is interesting. This is sort of an emerging dermatitis and just something we should know about.
Yeah, that's cool. Patton, any final comments before going to your first paper?
No, I haven't seen any autoimmune CAR T patients. I've seen lots of hematologic, but I don't think I've seen any autoimmune people.
What do the hematologic people get? Kind of a similar thing?
It's really nonspecific, pretty heterogeneous.
They get more cytokine release syndrome though, right? Like they can get sick and toxic. Like they made the point that LICATs was not seen in the hematologic.
Yeah, right. Because
malignancy treated patients.
Yeah, right. It's a totally different process. They get at this there's like a specific neuro reaction with the CAR T cell hematologic patients.
Yes, it is called immune effector cell associated neurotoxicity syndrome, or ICANS.
Yeah.
Yeah.
Okay. Well.
Yeah, it's just something different to be aware of.
Yeah.
Okay. Cool. All right, Patton, what do you got?
All right, my first 6-pack pre-proof article accepted May of 2025 in the journal Ocular Surface. We're all reading it. You guys know it. It's by Wynn. It's by Wynn et al. They just did their swimsuit issue. Unbelievable. It's by it's by Wynn et al. And it's titled Janus Kinase Inhibitors and the Treatment of Refractory Cicatrizing Conjunctivitis and Pemphigoid. Retrospective chart review performed at UT Southwestern Ophthalmology and Dermatology Clinics. Between 2015 and 2024, patients had cicatrizing conjunctivitis, were treated with a JAK inhibitor, had 6 months follow-up. Number of patients in the cohort was 32. Most of the patients, 71.9, had only ocular disease. The rest had a combination of ocular plus another skin or mucosal surface. Oral mucosa was involved most frequently. Most eyes had moderate to severe disease, with 8 eyes having only mild disease and 16 eyes having end-stage disease. Previously failed therapies listed at the bottom of table 1. Mycophenolate was the most frequently used therapy. Rituximab next most frequent, but still fewer than half of patients, which that's surprising to me. I try and get all of the scarring pemphigoid patients on rituximab just because, like, you don't want scarring to progress, so be aggressive. It would have been helpful if the authors explained corticosteroid use in more depth, like systemic corticosteroid. I didn't even know if all the patients were on systemic steroids. It was kind of confusing because one of the target is there was a complete remission and then complete remission off steroids, which one would assume they're all on steroids, but they didn't explicitly say that. So that was kind of hard to follow. So in any event, patient were placed on Berry or Tofa. It was about a 70-30 split. And they were followed for an average of 18.9 months. Figure 2 shows 2 swimmers plots. It's the same exact data. They just put it in a different order. So lots of different numbers in the results. I think kind of the takeaway points are they summarize them nicely in the discussion. 33 out of 34 patients had improvement after 6 months. 43.8, so a little under half of the patients with clinical remission at 12 months. Therapy was discontinued in 4 patients because of adverse events, infections. There was a TIA, there was a PE. 4 of the patients that achieved clinical remission or better relapsed. That happens a lot in immunobullous diseases, unfortunately. And over half of the patients had only a partial response. So JAK inhibitors seem promising. I'd rather use them than cyclophosphamide, which I kind of use in all my patients as well. Still probably going to use rituximab for ocular pemphigoid patients, but right. I mean, the difference would be maybe we start subbing in JAK inhibitors for the Cytoxan. Eyes that had higher...
Wait, the one cicatricial pemphigoid patient I have who, like, I don't even, like, when she comes in for her skin check, I'm like, hey, how are your eyes? Like, her eye doctor has her on Rituxan or whatever.
Yeah.
And it didn't, like, make her disease go away. Like, so maybe, do you think this will be second line after Rituxan?
Well, so what I do with these patients is I start them off on corticosteroids, moderate doses with the plan to taper over several months. I get them set up for rituximab and I start Cytoxan right away. And where I see JAK inhibitors coming in is maybe instead of the Cytoxan, I'm using a JAK inhibitor. Yeah, eyes that had higher Foster stages. So that's just like a grading, scarring, no scarring, mild scarring, moderate scarring, severe end stage scarring. So eyes that had higher Foster stages didn't respond as well. I think that is intuitive. Lower Foster stage. So the key is, and it is whether we're talking about JAK inhibitors or not, is initiating once you see scarring, jump on those patients, be aggressive. Initiating the JAK inhibitor in this particular study, those patients tend to do better. So it just reinforces the idea ocular pemphigoid needs to be diagnosed and treated aggressively. The biggest barrier is obtaining the medication at this point. They had to do prior authorizations. They got insurance to pay for it in a few patients. Patient assistance programs, which I don't quite understand how they did, because a lot of times patient assistance programs from the companies, it has to be an FDA approved indication. So I don't know how they did that. And then they had overseas pharmacies, which that sounded interesting. But yeah, I mean, small study, retrospective, but
a bad problem that's it's to the point now. People are going to need to start, because I was talking to Steve Davlui, who's like on the editorial board for JAD case reports. And he was like, we pretty much have decided we're not taking any more case reports about JAK inhibitors working for X, Y, Z. Like they work for everything. You can, if you're going to send in a case report, it's got to be that a JAK inhibitor didn't work. Like,
interesting.
It's just,
But these are, I think, sometimes helpful data for us to have, right? Like we're not going to get a JAK inhibitor cicatricial pemphigoid study. So, this is like important to have these things. And it's our only kind of leverage with payers sometimes. So I think that's right. Yeah.
It's going to be interesting to see how well they work in scleroderma. There was just a report of TOFA for morphia. I haven't looked at it yet to see, like, you'd have to have pretty bad morphia to like warrant TOFA. But it's going to be interesting to see, because I think of that as the other disease that just, it sucks. We don't have anything good for.
So that's like a fibrosing, like, it's not just an inflammatory disease, right? And like, do you think that there's some effect of the JAK inhibitors on the fibrosing or on the actual scarring, or do you think it's just the anti-inflammatory effect, or do we not know?
I mean, so we know that fibroblasts have the IL-4, IL-13 receptors on them, and whenever you block those, you downregulate, and those go through the JAK-STAT pathway. So yeah, I think that the JAK inhibitors, I would bet that they have some effect on fibroblasts as well.
And then Tim, because you treat a lot of these patients, is there any benefit to any sort of topical therapy early on, or is it like you just got to go systemic?
For scarring, no, like topical, and most of the good ophthalmologists know that. Like if they see scarring, they usually have them on some sort of topical, but they know, I mean, they're sending them to me because they know they need systemic therapy.
Okay.
See, I'm sending
even early on, there's no benefit. I guess they
Matt,
don't know they have it until they're scarring.
Yeah, Matt, you said the ophtho gives like I'm blown away by that. The ophthalmologists don't want to give like dapsone. They'll give prednisone, but it's the oddest interactions because patients come to me, they have nothing wrong with their skin. Even oral mucosa derms do, but they come in with eye disease, and I'm like, I think you're better. You look better to me, but you need to see the eye doctor to evaluate. And then they call me and they're like, we need to be more aggressive. And it's very weird.
Yeah. Ohio State has some, I think there's one particular ophthalmologist here who's like, that's his thing.
Okay.
Yeah.
No, and not that's not to say like UPMC optho people are terrible. They're really good. And where they do a lot of benefit is once we get the scarring quieted down, they do some pretty crazy like surgical procedures and flaps and things like that to correct entropion and scarring. It's pretty impressive. I don't know, like eye surgery.
Yeah. They're gonna start asking you to do that next.
Maybe I'll do it.
All right, let's move on to my. I'm going to sneak in two here. One was pretty straightforward, and it just confirms kind of what I thought. So it was long-term, real-world effectiveness of dupilumab versus upadacitinib in early treatment responders with atopic dermatitis. Results from the Central European Health Fund Registry. So this is a registry out of Poland. And participating in the registry is not optional. So if you are at like a hospital that has the Polish national whatever, your people got to go in this. So that 215 people who finished 40 weeks of Rinvoq and 220 people who finished 40 weeks of Dupi. And it was literally just, hey, from this date, the next 200 people who finished 40 weeks, we're going to get them. And the next 200 people who finished 40 weeks of that one, we're going to get them. So not a randomized trial or anything like that, but good, solid number of people. One of the big things I wanted to make sure of was that the people who went on Rinvoq hadn't previously been on Dupixent, and they hadn't, like less than 5 of them had been on Dupixent previously. So it wasn't like these were Dupixent non-responders. And the main takeaway was that the efficacy of Rinvoq was better early. So by 16 weeks, they were pretty equal. Other than whenever we look at EASI 100, Rinvoq had more people at EASI 100, statistically significant at week 16 and week 24. But by week 40, Dupi was statistically significantly better for EASI 75, EASI 90, and Rinvoq and Dupi were equal. And so because the real question has been like, we knew that because the data, AbbVie's been pretty careful to make sure their trials only go to 16 weeks so that we didn't know if Dupi was going to catch up or not. But sort of the expectation was, but you didn't know, did it catch up all the way or did it get close or what? And so this makes it certainly not a randomized trial or something like that. So I'm not going to sit here and say, well, this proves Dupi's better in the long term, but it shows me that the drugs become extremely similar in the long term. And then the second one was another Rinvoq one that was just very interesting. So this was a 24-week study out of Spain. I think they had like maybe 60 some patients in here. But the thing that was interesting, right? So great, Rinvoq worked just like it does and everybody else. The numbers were about the same. But what was really fascinating, they looked at diagnostic delay. And that is to me, that's one of, if not the biggest unmet needs in the dermatitis world, is from the day somebody first shows up at a doctor and says, I've got an itchy rash, until somebody says, oh, that's atopic dermatitis. How long is it? So their average diagnostic delay was 4.5 years. And I think it's even longer because I think there are a lot of people who never get the diagnosis of atopic dermatitis. They just don't onset. They don't have atopic comorbidities. It's not flexural. And so they're just called derm unspecified forever. So that it was interesting. This was the first data I've ever seen on diagnostic delay. And then second, and this was even more interesting, the longer your diagnostic delay, the less efficacy the Rinvoq had through 24 weeks. And so we know in alopecia areata that the longer you have it, the less likely you are to respond. But we really haven't had any data on that for atopic derm. And this suggests that the longer that diagnostic delay, at a minimum, the longer it takes to get the full effect of the drug. And that kind of makes sense because we do think of there as being the barrier function. Even once your skin looks normal and feels normal, its biophysical parameters are not normal. And it would make sense that the longer it's inflamed and damaged, the longer it's going to take for it to recover. Could also be that, the people with longer diagnostic delay had a different, means your atopic derm is weird. Maybe it's not really atopic derm. Maybe it's something else. So it's hard to ferret out exactly why, but two things there that were really interesting and that, to me help point even more towards there are a lot of these people who are just, we call them derm unspecified, and then they're stuck getting nothing but topical and systemic steroids. And not only is the steroid cumulative amount maybe causing some issues for them, it's also potentially also making their disease more recalcitrant.
So are you arguing that they're getting lots of topical steroids and now they're like totally destroyed their barrier and they're unsalvageable?
I don't know if they're unset because this only went out to 24 weeks. I'm wondering if it went out longer, if it went out to 40 weeks or 52, if we would get there, but harder to salvage is what I would say at this point.
So do you think that like this late stage, later in life atopic derm is the exact same disease immunologically as like the people who get it classic flexural as kids? Like, are you like, it's just the same disease? One person got it at 50 and one person got it at 4. You think they're two different diseases?
So I think that the final state of, because my definition of atopic derm is inadequate barrier function to resist normal environmental stressors. And I think the difference is childhood onset atopic derm, you genetically don't have a barrier. And when that you don't have an adequate barrier. When that happens early on, your immune system gets exposed to antigens in a different way, transcutaneous rather than transmucosal. And so then the disease takes a different course. I think the people who don't get it till they're an adult, and we know this, that genetically they're normal. They don't have the genetic stuff that the kids have. It's acquired through long-term exposure to chemicals in clothing, air pollution, laundry detergent, bathing your whole life, all of this stuff. So you end up in the same place with an inadequate barrier. But I think they were pathophysiologically different diseases because of my bet would be you don't have as much of a systemic Th2 shift in the adults. But I wish there was like two separate diagnoses I could make, like genetic atopic derm and acquired atopic derm, because I think they're different diseases, but they respond the same to therapy. And so from a practical perspective, we should just call them all atopic derm because then it opens up the therapeutic toolbox.
In the first paper that you covered, and I mean, I favor Dupi over UPA, but if I were to argue in UPA's behalf, did they break down 15 milligram versus 30 milligram dose?
They did, but it wasn't like they didn't. So they were allowed. Basically, the drug was used according to label. So the physician could increase and decrease and change the dose however they wanted to. My recollection is that the majority of people were on 30, but not all of them. But they could go to 30 if they wanted to.
Okay.
So it was, quote unquote, real world use, and that's an interesting challenge. So we've got an article submitted. Abro, Sabinco did a real trial like that, where like I was an investigator, and you could start about 100 or 200, you could switch anytime you wanted, and it gets really hard to report that data. Like, how do you know what I mean? We basically said, like, okay, 2/3 of people stayed on whatever they were on the whole time, and about both doses, if you started 100, there was a 1/3 chance you were going to bump them to 2. If you started 2, there was a 1/3 chance you were going to bump them down to 1. But it's a tough thing to figure out how to report that data. All right, next study. Ferris, what you got?
So I have from JAMA Dermatology, Zhao et al., ivarmicitinib for moderate to severe atopic dermatitis in adults and adolescents. So a phase 3 randomized clinical trial. I was not part of this one, but, always interesting. So what is ivarmicitinib? It's a highly selective oral JAK1 inhibitor developed to treat atopic dermatitis. And so, what does that mean? In vitro studies, they say, shows a 9-fold greater selectivity for JAK1 over JAK2. JAK2 is the kind of JAK that we like to avoid because it is what, inhibiting it is what increases the risk of anemia and neutropenia. So 16-week double-blind placebo-controlled study, 2 doses of ivarmicitinib, 4 and 8 milligrams, about 112 per group. Patients were 12 to 75. Typical inclusion criteria for an atopic derm study: BSA 10% or more, EASI 16 or more, IGA 3 or more, worst itch, numeric rating score of at least 4. And the primary endpoint was an IGA of 0 or 1. And so what percentage of patients got that? In the 4 milligram group, 36%, in the 8 milligram group, 42%, and in the placebo group, 9%. EASI 75, response 54% in the 4 milligram group. In the 8 milligram group, it was 66%, and it was 21% in the placebo group. So what was the mean? I like to kind of look at data sometimes like that. Placebo adjusted least square mean change and EASI score. Basically, how many points did your EASI score go down by? I actually like it better if it's percent, but it was like 6 points for the 4 milligram group and 6.8 for the 8 milligram group. 44% had at least a 4-point reduction in their itch. Safety wise, all of the adverse events of special interest were serious infection. There was 1 case of varicella, 1 case of sepsis in the 4 milligram group, 1 COVID-19 pneumonia in the placebo group. There were, other things that they saw. They saw typical kind of lab shifts that we see with JAK inhibitors, CPK elevations, LDL, triglycerides, and then decrease in platelets and neutrophils relative to placebo, transient and self-limited. They saw acne and folliculitis, particularly in the high dose group at 8 milligrams, mouth ulcers. There were no MACE events and no thromboembolic events. So, I thought it was interesting. Matt, I was curious because I think you think about JAK inhibitors all day long. What do you think about this, like relative to the other JAKs that we have at our disposal, and how important is this JAK1 selectivity?
So don't think it's important at all. So Sabinco is more JAK1 selective than this. Rinvoq is about 2/3 JAK1, 1/3 JAK2. Tofacitinib is primarily a JAK3 inhibitor. So, and these numbers, just their top line number of how many people got an IGA success or EASI 75 success, were almost identical to all the other JAK trials. So far, it's playing out that at least in efficacy terms, it doesn't seem to matter what JAK you block. Like JAK1, JAK2, JAK3. We don't know about tick in atopic derm. Nobody's really studied it yet. But JAK1, JAK2, JAK3 doesn't seem to matter. You seem to get the same efficacy. And whether there's a safety benefit to only doing JAK1, we're probably going to find out, because Incyte Povo, which is their JAK1 selective molecule that they're doing for HS, they're pushing the dose a little bit higher than we're used to in order to try and get more efficacy in HS. And so we'll see if that has a different looking safety profile. You imagine the higher they push it seems with all of the JAKs, the higher you push the dose, the more immunosuppression you get. But MACE and VTE, who knows?
Yeah, serious infections, the only ones that there was 2 in the low dose Ivor group. So, and then, it was interesting, like this isn't a huge study, but no MACE, no thromboembolic events. Now, when you put 12-year-olds in your study, that helps a lot too. Obviously, it's a little, versus if you look at an all adult population. So, but just thought I'd put it out there. I thought on people's radar, another JAK inhibitor.
Interesting.
Yeah.
All right. Patton, what do you got?
My second 6-pack was from the journal Social Science and Medicine and was titled Public Preferences for Skin Cancer Prevention Policies, a Discrete Choice Experiment in 3 European countries. It was by Boxbeld et al. Is available on...
How did you even find, did they come out with a swimsuit issue too? And that was the...
I can neither confirm or deny that. I will neither confirm or no comment. It came across my little search thing that I have
your
on
feed. Okay.
It's available online as of May 2025. The researchers wanted to see what sort of skin cancer. The headline grabbed me. I just kind of when I started going through the paper, I'm like, what did I do? The researchers wanted to see what sort of skin cancer prevention policies would be preferred by the general population of 3 countries: Spain, Netherlands, and Austria. They did this through an online survey using what is called discrete choice experiment, where respondents had to choose between 2 packages that had different policies matched with the effects and costs that the policies would have. An example is given in figure 1. And so if you look at it, respondents would basically look, and it was, campaigns, banning the sale of solar beds, banning tanning beds, a 30% price reduction in sunscreen, free provision of sunscreen in public spaces, and a free skin cancer detection app. So those were the options. These are the policies that we would institute. And then they would have theoretical, if this one decreased cases by 10%, this one decreased deaths by 15, here's how much it would cost. And you would just pick one of the 2, and then they would change everything, and you would go through that 12 times. So it's very a statistically sort of mathy driven way of, this is what epidemiologists do. It would be a blast to hang out with a bunch of epidemiologists, is what I took away.
Epidemiology Tinder, kind of.
Yes, right. Swiping left, swiping right.
Yeah, exactly.
It would be very,
but yeah,
very well studied Tinder. They would really know.
Over 2,000 respondents completed the surveys. The authors did a bunch of maths, and that's when I realized this was a huge mistake. They're tables with 50 numbers that have 5 decimal places, but you don't have to actually go through the tables, because they sort of make these other graphs that put that data in clearly. So figure 2A basically shows, it's a bar graph, additional taxes. That is the least favored attribute and the most important. That was kind of front and center. That's where the emphasis was for these respondents. How much is this going to cost? That was the same in all 3 countries. Table 4 shows what are called these MRS instruments. It's marginal
rate of substitution.
Yes. And so really what that is it's a number, and that's what patients say, I would accept this much in taxes for this particular product. So the top 3 measures in each country for which respondents would accept higher taxes were information campaigns, 30% reduction in sunscreen, and a free skin cancer detection app. Countries differed on what they would accept the highest increase for. Austria, information campaigns. Netherlands and Spain, it was the 30% reduction in sunscreen price. Netherlands, overall, a little bit cheaper, not to ditch our our Dutch listeners, but come on, guys. Overall, less willing to pay more in taxes for each of the measures. bans on the sales of tanning beds and tanning bed prohibitions were the least favored option in all 3 countries. I think that is kind of interesting, given, you look at what the American Academy of Dermatology and how they've had those campaigns. And I just, figure 3 shows medians and means of that MRS estimates based on the reductions of new cases and deaths. I mean, everything differs, but what stood out to me was the median. So like what most of the population feels. So it was like this panel B, most of the population don't seem willing to accept higher taxes if deaths were reduced. Like if they said deaths were reduced by 10%, most of the population, like the median, you had these outliers because the means did change. But for the median population, they're like, you reduce them by 10%, I'll pay this. If it's 25%, I still only want to pay what the 10% was, if I was reading all these graphs right. Figure 4 shows overall populations in all 3 countries did favor policy actions. It went up. So they asked them that question, do you favor government policies that would reduce skin cancers? And before they did this discrete choice experiment, majority of each country said, yes, we do. After it went up in all 3 countries. So, overall, if you're into the policy stuff, emphasis should be if you took this paper at face value, emphasis should be information campaigns, lowering the cost of sunscreens, minimizing the amount of taxes that would have to be levied to implement these policies, or don't increase taxes at all, and don't go after the tanning booths. That's the least favored sort of policy when you look at the populations.
So interesting. You think that'd be the cheapest thing, is to just say no more, like to ban solariums or like the tanning beds, right? Yeah. And I mean, that's like where most of our advocacy has focused through AAD, through like state organizations. It's not, I mean, it's really been on like not allowing minors to tan, people under 18, right? It's like why we don't let people under 18 get tattoos, right? Like you don't make great decisions.
It's almost like a like people like being said, hey, we encourage you to do these things that will help skin cancer. I just reflexively, it seems like they don't like being told what not to do. Bans, it's not like that's free, right? If you have to institute a ban and police it and regulate it, it's going to cost a little bit of money.
Yeah, you're right.
And that, there's this thing called the sun, which
what
may make banning tanning booths kind of silly.
Yeah.
So I don't know. I thought it was a cool paper.
I was going to ban that. That was going to be my campaign. Going to run for AAD president. I'm banning the sun.
Yeah, that was a Simpsons episode. Mr. Burns put up a big canopy over the whole city.
I had 2 main thoughts in this thing. Number 1, I would love to have known how they chose the countries. Like, because I do think of it as a good spread. I mean, I think of it as like, when I think of Spain, I think of as kind of laid back, kind of lazy-ish people, siestas and all that. When I think about the Dutch, I think about like very liberal, hey, let's smoke weed, let's have a red light district, woo. And then whenever I think about the Austrians, they're just like the Germans. So very like uptight and let's follow the rules and let's have rules, and the whole, like, if that's why they chose these 3 countries, was to try and get like a spectrum of stuff. Or
I like that. You've offended half of Europe in one.
Yeah, sentence. Right. Let's keep going.
Yeah,
right.
Let's keep going.
Maybe move it eastern. What do you say about the Indians?
But the other thing I thought was that these people, discrete choice experiments are always weird to me, because to get sunscreen to be 30% less, they basically, the mean taxes people were willing to pay was $70 a year. Just spend more. There's no way you're gonna save 70. If you're gonna pay $70 in tax, just buy the sunscreen at the price it's at now.
Yeah, right. It was like, we'll make it cheaper, but your taxes are going to be higher. Well, yeah, right. Just don't change my taxes at all, and I'll pay for that more expensive sunscreen.
Right. I did the math, and you'd have to buy like $600 a year worth of sunscreen before it became like economically beneficial to have the tax.
Yeah. If you were asking me, I would be like, I would accept $0 in a tax increase to make something cheaper. I would just don't tax me at all.
But the idea is that you're like you're spreading that over
Yes.
the
Yeah.
population, right?
Yeah.
So
Right. So you're
like,
helping the
yeah.
less fortunate.
I'm in this for me.
Okay.
Right on.
Yeah.
And I would pay a little to if they put free sunscreen in all the public places, so that when I got there and was like, oh damn it, I forgot to bring sunscreen, like there would be some sunscreen at the park or the beach or whatever.
I was surprised that one wasn't higher.
Yeah.
Yeah, it was
It
weird.
was a neat study.
It was. I was very interesting. It was. I was not expecting that banning the tanning beds was going to be like the least favored, because I feel like most people don't go to a tanning bed, so why would you care? Like, yeah, go ahead, ban it, whatever. I don't care.
Yeah.
It was weird.
Yeah.
All right, let's go on to my last one, which is a little bit kind of about an interesting thing of what's happening in the world. So these were the most recent North American contact dermatitis group results, the North American contact dermatitis group patch test results 2021, 2022. And the most interesting thing in this every year is like what's going up and what's going down, because we don't really actually have any good data on how often patch tests are relevant, right? So the vast majority of people, when you find a relevant positive patch test, they don't get better, right? That's what the data is. So it's hard to know, like, okay, are these actually meaningful or not? But the number of people with a positive test, that's a meaningful thing. So you can compare, is it going up or down? And the main takeaways were this: all of the metals, nickel, cobalt, gold, and chromate, from 2020, 2021 went up. So that suggests there's more metal sensitization going on. Maybe that's more tanning, or not more tanning, more piercing. Sorry. Benzalkonium chloride went up. So that is suggestive of more hand sanitizer use, because benzalkonium chloride is one of the ingredients in hand sanitizer. It's also used some as a preservative in some products. And then the other two preservatives that were interesting. So bronopol went up, which is a vaguely formaldehyde related one, but not much, not very formaldehyde related. Benzoates went up. Those are a replacement for formaldehyde and isothiazolonone. And so it leads to because then isothiazolonones and formaldehyde both went down. And so what's happening is they're taking the isothiazolonones and the formaldehyde out of products and putting in benzoates and bronopol and benzalkonium chloride. So allergy to those is going up. And that is the same thing that happened with isothiazolonones. They tried to get rid of formaldehyde by adding the isothiazolonones, and it turned out, oh my God, isothiazolonones are even worse than formaldehyde. So we're seeing a little bit of that same phenomenon happening. The other ones that were interesting, so 3 different non-sulfate surfactants. So oleopropylene, OPDM, dimethylamine, and ammonium and amidoamine, which are all markers for betaine allergy, went up. And so that suggests that the whole thing with sulfate-free products is causing an increase in allergy to other surfactants, because companies want to put sulfate free on the label, and they got to put something else in that makes it lather up. And then the last ones, some fragrances went up, some fragrances went down. Again, that's just expected, because like what's popular in a fragrance changes. So like, perfumes that people are buying now don't smell like your grandma's perfume. And so like some are going up, some are going down. That's expected. But then the other one that was very interesting, we just started patch testing widely to ammonium persulfate a few years ago, and it more than doubled, almost doubled. So it went from 1.9% positive patch test to 3.4%. And that is believed to be due to people using hot tubs more. So because about the only place people really get exposed to persulfates, it is the shock treatment used in hot tubs. And there's 2 main ways you can shock. You can use like a ton of chlorine, or you can use persulfates. When you use a ton of chlorine, like it makes it smell more chemically, and you got to stay out of it for like a day. When you use the persulfates, it doesn't smell chemically, and you can get back in it quicker. And so it looks like the persulfate allergy is rising because of both increased hot tub use, potentially somewhat related to the pandemic, and people just switching from chlorine to persulfates. The one pearl I would give from here is, if you get somebody who gets a new onset widespread sponge derm, ask them if they have a hot tub. And if they do, they need to stay out of the hot tub for like a couple months. If their rash gets better, they get back in the hot tub, and you have then confirmed that they have this persulfate allergy, and then they have to switch to persulfate-free shock for their hot tub. That's one of the most commonly missed causes of widespread dermatitis is persulfate allergy in hot tubs. Thoughts from you guys on it's interesting what's changing is the biggest thing that I always find with this.
I think it's interesting. That's good to know what's changing. How about the one that I think I mentioned it a few episodes ago, propolis from
Yes.
What's that doing?
So it dropped precipitously because it was like ludicrously high. It was like at 10%. And that like there was like no way that has to be false positives, and they think it was that the allergen that was being used was like too strong or something weird about the allergen. So propolis went from 8.7% down to 2.2%. For our listeners, propolis is an allergen in beeswax, and it's one of the things if you see somebody you think has allergic contact dermatitis of the lips, you want to make sure that they use nothing but either Vaseline or CeraVe healing ointment. The two things you do there, you switch to nothing but those two things on your lips, and then you switch to a flavor-free toothpaste, because those are that's almost all allergy for lips are those three things: flavor and toothpaste. Favorite toothpaste for that, by the way, is Cleure, Cleure. And then making sure that you don't use anything except for CeraVe healing ointment or plain Vaseline on your lips.
Is the sodium lauryl free sulfate SLS free toothpaste, is that a thing for lip derm or no?
No.
Okay.
So SLS
sodium lauryl sulfate.
Yeah, nobody's allergic to SLS. It can be irritating. So there are some people who get like irritant, like they'll get oral ulcers or oral pain from SLS.
Okay.
But it doesn't cause it's not allergic contact dermatitis on your lips.
Okay, that I must have been thinking of the oral ulcer stuff. Yeah,
but there is some suspicion that eosinophilic esophagitis may be partially caused by sodium lauryl sulfate in toothpaste, because now whenever you brush your teeth with sodium lauryl sulfate, you swallow some of it, and so now you're getting the sodium lauryl sulfate that's breaking down the protective lining of your esophagus, and that makes you susceptible because something happened, right? Eosinophilic esophagitis didn't exist when we were medical students.
I thought it was because we have Dupixent that we needed another disease for.
That's what they
diagnosing it. We all
needed to come up with.
It's like restless leg syndrome or whatever that like never used to be a thing until we had a drug for it.
So we had a drug, then it's a thing.
So it's eczema of your esophagus. All right.
Yes, that's how I think of eosinophilic esophagitis is eczema of your esophagus or asthma of your esophagus. You could think of it that way too. But yeah, so that's what's the hot new thing in the contact derm world, were these results, and it's definitely interesting. The ammonium persulfate's the main clinical pearl to take away from the whole thing. Good.
When I think hot, I think the patch testing contact dermatitis world. So that's good. Thank you.
It's as popular as the swimsuit edition of Ocular Surface.
Exactly.
Here's a picture of the eye. You can't see the swim, but we promise they're wearing one.
They're
wearing one. All right. Well, that was a great 6-pack episode. I want to thank everybody for joining us. If you've got questions, comments, topics you think we should cover on the show, shoot us an email at questions@dermsanddrugs.com. We hope you learned a few things. We hope you laughed once or twice, and mostly we're hoping you're planning to join us next week. Until then, I'm Matt Cyrus.
I'm Tim Patton.
And I'm Laura Ferris, and we are Derms on Drugs.